Hey guys, welcome back. Um, this is lesson 7 on stems and roots. Hopefully, you guys have tried the summative quiz last Friday. Again, there's no rush on that. If you need some extra time to look over your stuff, no problem. Please be aware that the quizzes are going to be quite a bit smaller in the future. So the reason why the quiz was so big was that that was the first opportunity I was actually allowed to do an assessment from any of the online modules, and so a lot of modules had built up previously for me to assess. But now we're just going to go week by week, so more or less. So this the quiz that's coming up on this Friday is going to be based on last lesson, which wasn't included on the previous summative quiz, and it's also going to include today's lesson on stems and roots, and then Wednesday's lesson on transport in plants. So, and then we're going to do a summative quiz, and then the next week is just going to be on its own. It'll just be the Monday and Wednesday lesson, and that's what's on the summative quiz, and they're not cumulative. So, just so you know, that's how it's going to work from here on out, and we're not going to have stuff being cumulative on and on. Which I hopefully will be less stress for you, that you don't have to go back and review your notes from like a huge volume of items. Okay, so from the previous lesson, we left off with some analysis questions for you to think about. I'm just going to discuss these very quickly before we get into stems. So one of the questions here was this idea that you're looking at a leaf from a desert environment and one from a rainforest. So what are some potential differences you'd see in the leaves that would make it more adapted? So the big difference here is the amount of water, obviously, that's present. Desert being very low water and the rainforest being very high water. So one of the adaptations that leaves have in general is this idea that they have these holes in the bottom, the stoma, which are protecting, which are allowing gas exchange to occur, and they have those guard cells that are around the outside that are opening and closing the hole. So in real life, one of the major adaptations that's seen is that how the guard cells actually open and close is altered, or the number of openings is altered. So some desert plants have far fewer of these openings in them, because keep in mind that those holes are just opportunities for water to evaporate into the leaf. Water can escape through the holes. So you would think that a plant that you'd find in a desert environment would have fewer of those. There's also going to be a lot less photosynthesis going on in the desert environment, so there's less need for gas exchange, so you need fewer holes for gas exchange. So you're probably, either way, you're going to find fewer of those stoma in a desert environment. And then the other cool adaptation is that is when these open. So in typical plants that are in moderate or high moisture environments, like basically all of the plants that you would find around here, they're going to have their stoma open, or their guard cells will open the stoma when there's water available. So anytime there's water available, they're going to open, and then that's going to allow photosynthesis to occur. In desert plants, there are some other considerations, which is when do you want to do gas exchange? For desert plants, it actually is probably a better strategy for them to do gas exchange at night. So what happens is their guard cells often will open up and allow gas exchange to occur when there's the least opportunity for evaporation to occur of water, which is at night. So they'll open up at night, they'll allow gas exchange to occur, there'll be some O2 that goes out. I should reverse that. There'll be some O2 that's going out of the leaf, there'll be some CO2 going into the leaf at night, where there's going to be low likelihood of water escaping through the stoma, and then during the day, the stoma or the guard cells are going to close the stoma and not allow any gas exchange to occur. That does inhibit the amount of photosynthesis you can do, because the CO2 will build up, the O2 will build up inside the leaf and actually prevent that chemical reaction from going forward. So it does limit the amount of photosynthesis that you can have occurring. But On the positive side, it prevents a huge amount of evaporation. So regular plants that are living in moderate to high moisture environments are losing a ton of water through evapotranspiration, which is water that is exiting through the stoma. We're going to talk about that in the transport lesson in a lot more detail. That's coming up on Wednesday, because that's actually pivotal for how water is transported in plants. But anyway, so that'd be a major difference that you would see between those two. Okay, the idea of eating your leafy green vegetables. So what do we need that for? So that component of your diet is fiber. That's why you got to eat the leafy green vegetables. And the fiber is cellulose. That is the key. You need cellulose cell walls, which are found in large quantities in leafy green vegetables, because that fiber, that cellulose, holds onto water content in your digestive tract and keeps everything soft and mushy so that it can proceed through your digestive tract and out the other end. If you remove the cellulose component, that water absorbing cellulose, oftentimes the contents of your digestive system can get really dried out on the route out, especially in the large intestine, and then they can become very hard and difficult to transport. And that causes constipation. So, and there's a bunch of side effects of having that chronically in your body. You have high pressure that occurs in your abdomen because you have to push in order to remove waste, and that can cause all kinds of other problems. When we get into the body systems unit, we're actually going to talk about the digestive system and some illnesses of the digestive system in a little bit more detail. And then we'll come back to that idea later. But anyway, cellulose is a key component of the human diet. You have to eat at least a moderate amount of cellulose. Most North Americans don't get enough because you need that those cell walls basically in your digestive system. Okay, and then lastly, the idea of this sub-zero temperature adaptation in angiosperms. Well, what do angiosperms do? Most of them drop their leaves when the temperature goes below freezing. So most flowering plants will drop their leaves, or some of them just have their stems shrivel up. They pump all of their nutrients that they can into their roots, and then they just allow their stems to shrivel up. So a lot of monocots have that adaptation. And you can, if you take a little stroll through a forest right now, you'll see that there's like all these dead small plants from the previous year because even their stems have dried up and fallen off. And then that allows them to cope with below freezing temperatures. If you have a woody stem, if you're a dicot, which we're going to talk about today, your adaptation is usually not to let your stem die, but just drop your leaves so then the leaves don't get destroyed. Or you don't have to at least repair the damage caused by frost. All right, let's get into today's lesson. So today we're going to spend some time talking about stems. We're going to talk about this idea of vascular tissue in the stem, where it's placed, what it's used for. So the purpose of those stems is to connect the root and the shoot system. So the root system is often used for storage. It's where the water is going to come from. And then that shoot system is what's going to be producing the leaves and the flowers and everything else at the top. And so the stem has got to connect those two together. If you're making nutrients at the leaves, you got to send it down to the roots. If you are doing photosynthesis at the leaf, you need to bring water up from the roots to the leaf because that's where it's coming in. And so we're going to talk about sort of like the basic features here of the stem, how it's organized. And then on Wednesday, we're going to get into the details of how stuff actually gets moved around in these cells. That is how water gets pumped around or moved around, and how sugar and other nutrients is moved around the plant. The structure of the xylem and the phloem. In this lesson, we're going to talk about where they're placed in the stem and in the root. And in the next lesson, we're going to get into detail about how they function at like a sort of a cellular level. We're going to talk about the idea of woody or herbaceous stems. Those are the two basic kinds of stems, and you've seen them outside in the world before. And we're going to talk about the arrangement of those vascular bundles. Vascular bundles are xylem and phloem. And then we're also going to talk about this idea of production of new xylem and phloem. So how that works in plants, where they build their xylem and phloem, and then how it can be potentially cyclical. Okay, so we mentioned here that the idea here is that it's a connection. We're going to build a connection between the leaf and the root, pumping water up in general, and pumping sugar in general, pumping sugar down. It really depends on the season for sugar, but it's usually being pumped down to a storage area. Another really important function for the stem is to raise and support the leaves and the reproductive organs like flowers, but you got to get the leaves up off the ground because you're competing with other plants for sunlight, and the higher you go, the better you are at claiming the sunlight for yourself. So having a long stem is potentially very advantageous to a plant. So it's also serves as a support structure, which maximizes its exposure to sunlight. You also want to raise your flowers up. So if you want to get insects to notice your flowers, you got to get them into view. So you got to get them off of the ground, or at least it's beneficial to get them off the ground, and potentially when you distribute seeds and things like that, or distribute fruit, if that's part of your reproductive strategy, you want to raise that stuff off the ground as well, so other animals can find those things if that's part of your reproductive strategy. It is also possible that the stem could be modified to store water and carbohydrates. There's a question that you're going to look at for homework where you're going to look at sugarcane and look at where potentially sugarcane stores its carbohydrates. Okay, so let's start off with the basic structure here of stems. There are two main kinds. So the herbaceous stem is basically green-stemmed plants. So if you go out into your garden or go out into the woods and you find like a green-stemmed plant, what are some examples of a green-stemmed plant? Corn would be a really good one. Hostas. Maybe you have some hostas in your garden. Do you know what those are? Let me see if I can do a real quick Google check here for hostas. They look like this. These are hostas. And they have green stems. Let's see if I can get a good picture of their stems. Not the best. They're green stem down there. All right. So like plants like that. So you'll notice those plants have something in common. They have parallel veins on their leaves, which means that they are monocots. So herbaceous plants are monocots. And if you'll remember from the last lesson, monocots are plants that have one single leaf that comes off of their seed when the seed germinates. Okay, and they are a subtype of angiosperm or flowering plant. So monocots have herbaceous stems. Dicots, which are the type that have two seed leaves coming off their seed when they germinate, have woody stems, as do gymnosperms. So gymnosperms are plants that produce cones. You would call them conifers, plants that have needles, basically. So both of them have a woody stem. So a woody stem is stems that have wood in them. And that makes up a huge proportion of plants. Okay, what has a woody stem? Like a maple tree, or like a carrot. A carrot is weird because it has all the structure of a woody stem, but it only ever gets a single year of growth. So when we look down at the actual interior structure of the stem, carrots stems will look like this. They are green in appearance. They don't have bark on the outside or anything like that. But they are physically, like, they have all the characteristics of one of these dicot stems. What else would be? Excuse me. Like a birch tree. Like anything that you would think of that would potentially have a woody stem, like that would have bark. So all of those things would fit under a dicot or a gymnosperm woody stem. So those are the two basic kinds. And those stems have vascular bundles in them. Vascular bundles, just like the vascular bundles in the leaf, have xylem and phloem tissues. So if you haven't filed away what each of these does yet, you're really going to want to do that because it's sort of like a key piece of information from this course. So The xylem is responsible for water transportation, and the phloem is responsible for nutrient transportation. It's mostly glucose that's produced through photosynthesis, but there's other components to it as well. So, again, xylem's water, phloem is glucose, and they are usually found together in bundles, where one side will be the xylem and one side will be the phloem. When you're looking at a cross-section of a stem, the xylem is always the closest to the center, and that's in every type of plant that has vascular bundles. So monocots, dicots, gymnosperms, the xylem is always the closest to the center of the stem in the vascular bundle. The phloem is always closer to the outside. So if we look at monocot and dicot stem below here, so this is a dicot stem, there is a layer around the very outside, the epidermal layer. And in the previous lesson, you would have read about epidermal tissue. It forms, it's like, dermal tissue rather in plants, which is similar to epidermal tissue in animals, or epithelial. Oh boy, I'm confusing terms here. Epithelial tissue in animals provides a protective covering. So the epidermis in plants is dermal tissue in plants, and it protects the outside. That would be where the bark is forming. So the bark would be the epidermal tissue. Then below that, you're going to see the vascular bundles. So the vascular bundles are right. Let's see if I can highlight it here. This is a vascular bundle right here. And then on the outside of the vascular bundle, there's... Oh, that's going to be too big. Let's try this. On the outside of the vascular... why did I use blue? That's hardly even visible here. Let's use orange. On the outside of the vascular bundle, so towards the outside of the stem, that's going to be the phloem. So I'll add that in here. And towards the inside, people see blue. What would you be able to see? Red? Sure, that'll work. The inside here, this is going to be the xylem. So xylem always on the inside transports water. Phloem transports sugar on the outside. In dicots, the layer in between those two layers, and it's really, really hard to see on this one. We're going to be looking at a better diagram for this later, but there is a little layer that runs right through the center right here. And that layer in the middle is called vascular cambium. Vascular cambium is the layer that produces new layers of xylem and phloem. So during the growth season of the plant, it's going to produce brand new xylem, which are going to stack on the outside of the old xylem and push the old xylem towards the center of the tree. And it's going to create new phloem on the outside as well, which is going to spread apart, and it's the reason why the trunk of the woody stem gets greater in diameter year on year. Okay, so there's a layer right in between the xylem and the phloem. That's the vascular cambium. It's a little more obvious in the other diagram that we're going to look at. So this is a dicot stem, and you'll notice that the vascular bundles are all around, excuse me, all around the outside of the stem. So here's another vascular bundle. 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 So they're in a ring around the outside in a dicot. The center is usually some type of sclerenchymal tissue, parenchymal tissue. It's basically structural tissue that's found in the center. You call it wood. So the old xylem tissue, that's the old tissue that used to transport water, gets continuously compacted into the center of the stem, and it so as the new tissue forms, the new xylem tissue forms, the old xylems get pushed in towards the center and they become wood. So xylem tissue is actually dead. The cells are dead when they're functioning because they're basically hollow tubes. We're going to get into this more on Wednesday. And so those old hollow tubes, when they get compressed and pushed into the center, they become the heartwood of the tree. Okay, so you got heartwood in the middle, which is a really tough parenchymal tissue. Whereas in a monocot stem, so this is a monocot right here, you'll notice that the vascular bundles are actually all around. So we've got a vascular bundle here, we got a vascular bundle here, we got a vascular bundle here. They're distributed throughout. And within each vascular bundle, you'll notice that it still follows the same trend, which is that the xylem is towards the center and the phloem is towards the outside. So I'll show you an example here. I want to color code it the same. What did I do for xylem? Is that pink? Red? So the xylem would be these. Oh. Right here. Oh. I'm struggling. Not that. Whereas the phloem are towards the outside. And if you look at any of these vascular bundles, the arrangement is the same. So you can see it over there as well. So we've got the phloem on the outside, and we've got the xylem on the inside. So how can you tell the difference between xylem and phloem? Well, the xylem cells they look hollow. They look like big empty tubes. You can kind of see it here a little bit. We're going to look at it in more detail in a second. Whereas the phloem tissue, the cells are a lot smaller because they're living cells that are pumping things, as opposed to the dead xylem tubes, which really are just empty tubes. Okay, so the basic way that you can identify monocot and dicot, which are both angiosperms, they both are flowering plants, is that in a dicot you get your woody stem with bark on the outside, and then you've got that ring of vascular tubes that goes around the outside with xylem and phloem in it. In a monocot, you have there's still something like a bark on the outside, an epidermal layer, but it obviously is not as thick as bark. And then you've got a distributed system of vascular bundles that are all over the place. I should also mention that this applies, the dicot stuff also applies to gymnosperms as well. So gymnosperms are conifers, right? I'll just draw like a little picture over here so you can see a gymnosperm. So gymnosperms also have this same configuration for their tissues. Okay, so that's the basics of where you're going to find the key tissues on the inside. Let's talk a little bit about this idea of woody stems. So as I previously mentioned, the outside of a woody stem is going to have xylem. The xylem is towards the inside. So, and you'll notice it's layered here. So we've got, this is the outermost layer. This would be the most recent layer of xylem. And then all of the previous layers of xylem have been compacted towards the center of the tree, which is going to form the heartwood. So the last few layers of xylem is considered to be the sapwood. That's towards the outside, and then as they get compacted, obviously they're not functional anymore. The tubes get totally compacted, and then they become the heartwood in the center of the tree. Because you got heartwood in the center, then you got the xylem tissue, and then I mentioned that there is a layer in between called the vascular cambium. So this layer right here is what grows new xylem and new phloem. So it's continually moving outwards as it produces xylem behind it, and then on the outside of the vascular cambium, it's producing new phloem. So the phloem is the part that is pumping nutrients, and those are living cells. And they don't get compacted at the end of the season. You need to make new phloem because the trunk is getting wider, and so more cells are necessary to fill in all the gaps as it gets wider. But it's not like the old phloem die and get compacted like the old xylem do. And then on the very edge of that, you've got cork cambium. Cork cambium is just what produces the bark. And then beyond that, you have the actual cork, the bark itself, which is on the outside. And it serves as protection for the tree. So why is bark important? It's protective. Mostly for preventing insects from stealing nutrients from the tree. So it's for preventing incursion from animals and plants. So there are types of plants as well that will grow into the outside of another plant and try and steal the nutrients. So keep in mind that these woody stems are keeping all of their nutrient transportation, that's the phloem, all just around the outside of the plant. So if you were to damage the tree just underneath the bark, where the phloem is located all the way around the outside, you're going to end up girdling the tree. Girdling the tree means that you've cut all of its nutrient transportation all around the outside, and that's fatal for a tree. So that's, if you've ever heard of the emerald ash borer before, the emerald ash borer gets into the phloem, it digs down into the phloem of ash trees. That's why it's the ash borer. So it digs down through the cork into the phloem layer. So it comes in here, and then it digs a circle through the phloem all the way around the entire ash tree. So once the emerald ash borer gets through the entire phloem all the way around the outside, the tree no longer has any way to transport nutrients around the tree. So then that is fatal for the tree. So the cork obviously has an important function here, in that it generally prevents the incursion of insects, but some of them can get inside. The same thing is true, by the way. Sometimes you'll see people that will like cut the outside of a tree for whatever purpose. It's not such a big deal. For example, if you're collecting sap for the purposes of making maple syrup or whatever, you're only cutting a little bit of phloem, not a huge amount, and so then that can grow back. There's lots of other phloem to support the functioning of the tree. But if you were to cut a gap all the way around in order to harvest something like maple syrup, it would kill the maple tree. So you do need to be careful how much you actually cut around the outside. The same thing is for putting nails and stuff like that into trees. If you damage too much of the phloem around the outside, it's fatal for the tree. So this brings us to the idea of a growth ring. So I'm sure you've seen growth rings in a tree before. In grade 10, you may have even looked at them as part of the climate change unit. You may have looked at dendrochronology. So actually, some of this might even be familiar to you already. Here's a blow up of a woody stem from a tree. And you'll notice that here's the vascular cambium right here. So this part right here is the part that is forming new growth for the tree. This little strip right here. And it's creating new xylem for transporting water over here as it goes. So it created these cells here, and that created these little cells here. And at the very beginning of the season, when there is a ton of water present for the tree, or for the dicot or gymnosperm, it produces really big xylem cells because it's got lots of available water, and so it has a capacity to produce these very low density, giant water transportation vessels early in the year. And so that's often called springwood, because the spring is generally the time of year where there's the most water available. And that's like the light colored wood in the tree ring. Then towards the end of the year, as the water starts to become more and more scarce, the xylem that's produced tends to be much smaller. And then those much smaller xylem cells are darker in color because they're much more compact and more dense. And so that's often called the summer wood. And whereas in the winter, the fall and the winter, there's pretty much no growth happening in the tree. And so then there's no xylem being produced during that time. So we've got spring growth and summer growth, and then that gets cycled over and over again. So if you look back into this tree's life, you'll notice that there is a previous layer right below it that has some summer wood and some spring. And what is probably another layer here of summer wood and spring. So this tree is probably 3 years old. It's in its 4th year, or it's in the winter of its 4th year, probably. It's in the winter of its 3rd year, so it's about to start its 4th year's growth. The layer that's right in the center here, this part over here, is not old xylem tissue. It's often just like it's basically where the stem was of the original tree, which has parenchymal tissue. It's like structural tissue in the center of what it would have been the center of the stem. So that's towards the center. In some trees, the xylem tissue really gets compacted towards the center and forms a really hard heartwood in the center, but that's not what this tissue is right here. So this is like sclerenchyma or something here in the center, or it would be parenchymal tissue. Some plants, some dicots store energy in that tissue in the center. It's like a spongy, soft tissue. Anyway, so that's where the idea of growth rings come from, and we can use that to date trees. We can use it for dendrochronology, which you probably talked about in the climate change unit in grade 10. And so that all comes together with this idea of the structure of dicot stems. So you can do that in dicots. Dicots do this. Anything that has that veiny pattern on the leaf, remember that those are dicots. So if you've got a leaf and it's got veins in it, that's a dicot. And then so they're going to form some type of tree rings, or some type of rings in their stems, so you can age them. And the same thing is true for trees that have needles. So if you've got a needled tree, both of those are going to have these tree rings that you can use to identify their age. Monocots, things like corn and stuff like that, they usually don't have multiple years of growth on a single stem anyway, but they would have distributed, even if they did, they would have distributed vascular bundles, so that you wouldn't see tree ring growth. Okay, so that's the basics of stems. So there's some basic idea about the structure and function of the stuff in stems. We're going to get into a lot more detail when we talk about transport in terms of how things are actually moving through these structures. But for now, that's good enough. That's just the basic structural stuff. I'm going to spend a few minutes talking about roots, not in as much detail as the stems, because the basics are all the same, in that they've got vascular bundles, they've got some structural cells, and then they've got an epidermal layer around the outside. So the basic structure stuff is the same. They have a slightly different function. They're used to anchor the plant in general. They absorb water and minerals from the soil. And for most plants, they're used for storage. So they can store nutrients as simple sugars, or more often as starches, which are long chains of simple sugars bound together. And the the structures of monocot and dicot roots are a little bit different. We're going to talk about those differences, what they have that makes them identifiable. And then we're also going to talk about a little bit about how gymnosperms are similar or dissimilar from the others. So, as previously mentioned, as I just mentioned, they're used to anchor the plant and keep it upright, which is important, especially if you've got a really big tree. You gotta keep that thing upright. There's a lot of wind and stress that's pushing on the plant to try and knock it over. And they're absorbing water and nutrients from the soil. And they have a storage function. Again, we mentioned all these things. Okay. So what are the two basic kind of roots then? Well, one of them is called a taproot. So the taproot is when the main body of the root goes straight down from the plant, like this. And then there are some sort of side roots that grow off of that main taproot. So like the best example I can think of is a carrot. You guys have seen a carrot before. A carrot is a perfect example of a taproot. Okay, a root that goes straight down. A carrot is a dicot. Again, that one's a little bit weird too, because it doesn't really have a woody stem. At least it doesn't appear to be woody. It has the structure of a dicot stem in that it has the ring of vascular bundles around the outside. And if you ever look at a carrot leaf, it has a veiny leaf. So dicots have these taproots. This also makes sense when you think that a lot of dicots are really tall. All trees are tall, and a taproot is excellent at supporting a very tall very tall structure. So dicots have this root configuration, as do gymnosperms. So those are conifers. So both of them have a taproot structure for anchoring them in the soil. Monocots, on the other hand, monocots tend to always have fibrous root systems. So fibrous roots are roots that kind of just go out all over the place from the plant. They're great at preventing soil erosion. They're not so great at supporting really big vertical structures from the plant, but they're great for keeping a plant from secured into soil. So monocots have fibrous roots, dicots and gymnosperms have taproots. The other quick thing I want to mention is if you look down at the cellular level at the root, just like the stem, they have an epidermal layer around the outside, and it serves as protection against insects, just like the epidermal layer does in the bark. And they also have these single cell root hairs. So the root hairs are growing out of single cells that are in the epidermal layer, and those root hairs are increasing the surface area. So obviously, if you make a cell really, really long, like this, and then maybe you have your nucleus and stuff down here, that's going to increase the surface area of that cell. And the epidermal layer has one of the primary functions of the epidermal layer is to absorb things from the surrounding environment. You want to absorb phosphorus and nitrogen and all kinds of things from the surrounding environment. So in order to get the maximum amount of absorption, you got to increase your surface area. So the root hairs are on all types of plant roots, and they're there to increase the absorption. So below that is a little bit of structural stuff. The cortex here is structural. And then in the center of the root, that's this part right here. That's where you're going to get your actual vascular bundles. So you've got xylem and phloem in there. In between, there is a little layer of cells called the Casparian strip, or I should say they're endodermal cells, which are just structural cells. There's nothing in particular that's super important about the cells themselves, but except for the fact that they have this strip around the outside. So the Casparian strip is basically a seal. So it's watertight. You you want to have a barrier for water. Water is just going to move due to osmosis, right? So H2O can just kind of make its way just through osmosis into this tissue right here. So if the root is sitting in water, it can just pump the water will just naturally flow from high concentration to low concentration into that cortex around the outside. But you don't want it to flow directly into the plant. You want to control the entry of water, and that you also don't want things to flow out of the plant. So for example, if it's really dry outside the plant, you don't want water just flowing out of the plant into the dry soil. So in order to prevent that stuff, you got to have a spot where you have a gate, basically. You're going to allow water in at this time. You're not going to allow it in at other times. Maybe you're going to pump water in through the cell at different times. And so that's what this endodermal layer does around the outside. They're pumps. They pump stuff in and out, pump in nutrients, pump out nutrients, nitrogen compounds, phosphorus compounds, sulfur compounds, and pumping water in and out. And in order for them to do that, they have to be sort of sealed. So unlike the other cells in the cortex, they have this strip, the Casparian strip, which is basically like it is a seal. So their water and stuff cannot naturally go in between these cells. I think I was meant to write that over here. Oh no, the function of root hairs. So root hairs, I'll just add that in. Whoops. Dramatically increase the surface. I'm having trouble typing the surface area of the roots. Which allows them to increase the amount, the rate at which they bring in nutrients from their surrounding environment. Okay. So last thing I'm going to mention here about roots is where you find these structures within the roots. So we talked a little bit about structure here of the outside of the root. That's actually the same for monocots and dicots, in that they have that Casparian strip around the outside and a little bit of cortex and root hairs on the very outside. So that is true for both of these. You would find that right in the outside of both of these. So they've got that epidermal layer on the outside right here, and then those cells have little root hairs, and then again that's found on both. So you'd see that here too, with little root hairs, and then you're going to find that cortex tissue that's right here, or on this plant it's right here. So there's more cortex tissue here in the dicot, but both ways there's cortex, and then the Casparian strip is going to be found right here on these. That's where the endodermis is. Okay, but what is the main difference here between these two? Well, this one over here, A over here, is a monocot. And monocots have a circle of xylem and phloem around the outside. So here we've got real big xylem. Remember, those are empty, dead, and they're for transporting water. And then you've got the phloem, which is tucked always towards the outside, so it's exactly the same down here, and this is where sugar transportation is going to happen. And so that is these cells here that are towards the outside. So here they are here. So the sugar transport is towards the outside, and the water transport is towards the center of the plant. So they're in a circle in the monocot, and then in the dicot, oh, that's not how you spell dicot. The dicot and the gymnosperm, the vascular bundle is quite a bit smaller. And then you're going to have, always towards the center, you're going to have your xylem, which are the big empty dead cells for transporting water, and then the smaller phloem cells here, which are used for transporting nutrients towards the outside. So if you see an X of phloem in the center, or sorry, not a phloem, of xylem, an X of xylem in the center, or a T, that's a dicot stem. So that's the main way that you can tell the difference. So if you see that little T structure in the middle of xylem, that's for a dicot or a gymnosperm. But if you see a ring of vascular tissue around a cortex in the middle, or parenchymal tissue in the middle, you're going to see that is for a monocot. Okay, so that's the basics of the structure of the root and the stem. What I'm going to get you guys to do is to do some of these little analysis questions for homework. So these are questions that may require you to do just a little bit of research. Some of these I actually may have answered in the lesson. I think I talked about what the core cambium does already. And then I'm going to take these up at the beginning of the next lesson. But you should do them yourself, do a little bit of research. And we'll chat about this at the beginning of the next lesson. Once you've completed this stuff, try the questions in the exit quiz. And then that's all there is to do for Monday. And I will see you guys on Wednesday, where we're going to talk about transportation mechanisms in the plant in a little bit more detail. See you then.